Chapter 22, Topic 22.4, Using Microorganisms to Make the Things We Need. The objectives for this topic are: state the general aims of industrial microbiology, list 5 different types of substances produced from industrial microbiology, and their applications. In this topic, we're going to cover industrial microbiology, from microbial factories to industrial factories, and substance production. Industrial microbiology is using microorganisms to make products on a large scale. This leaves out home hobby brewers, but not commercial microbreweries, to give you an idea of what we mean by large scale. These two tables show a very small sample of the industrial products and enzymes produced by microbes. Isn't this impressive? Doesn't it make you like bacteria and fungi a little more? Microbes, specifically bacteria and algae, are being used to solve our energy problems. We are trying to develop bacteria that will ferment corn husks and stalks into ethanol rather than having to use sugar-rich corn kernels for ethanol production. Algae, like those in this photobioreactor, produce oxygen and oil. If we can figure out how to do this on a large scale, in a cost-effective way, we won't have to drill for oil. And the algae will suck down the CO2 that is released into the air from burning the oil. It's a win-win situation. And, if you've ever been downwind from an oil refinery, the scent has got to be better. I imagine it would smell like either pond scum or cooking French fries. You know, the refining part of the algae oil? You heat up the oil to refine it. Cooking French fries has hot oil. Yeah, it was funnier in my head. Microbes are living factories. Depending upon the type of microbe, they make just about everything that you can make in a chem lab, and they do it more efficiently. The trick is keeping them happy and healthy in a human factory setting. You have to keep them in the proper phase of the growth curve. For some products, the microbes need to be kept in the log phase. For others, like penicillin production, the fungi need to be kept in the stationary phase, which is when they make the most penicillin. When they start running out of food, they think bacteria are outgrowing them in their native soil environment, so they release penicillin to kill the bacteria. Cool, huh? Some microbes, like the yeast that make our alcoholic beverages, need to be cut off from oxygen at the right time to switch them from aerobic respiration to fermentation, and on and on. This figure shows an industrial fermentator. Nutrients and microbes are added here. A water jacket is used to keep the fermenter at the correct temperature. Air is bubbled in at the bottom of the fermenter so the microbes that must use oxygen to make their product have enough. The harvest line routinely removes the product and microbes, of course. The microbes are removed from the product most of the time. You want to be injected with pure, sterile penicillin, not the penicillium mold. There are sensors and a port for drawing samples, so you can check microbe nutrients and the concentration of the product. All of this needs to be done in the most sterile conditions. You don't want to contaminate the product with pathogenic bacteria or non-pathogenic fungi, or microbes that will compete with the hard-working factory pure culture. Food factories generally have less stringent requirements than pharmaceutical factories, like this one in the picture. It makes sense. You need a lower number of microbes to have an infectious dose for parenteral entry than for gastrointestinal entry, most of the time. Well, that's it for this topic. Here are reminders of what the learning objectives are. State the general aims of industrial microbiology, and list 5 different types of substances produced from industrial microbiology and their applications.